Okay, I'll talk a little bit about our TSTS project that started last summer at the StREAM lab, focusing on the fate and transport of antibiotic resistance genes. And this is a collaborative project with myself, Cully Hessian, Leanne Cromides, Catherine Knowlton, and Kang Sia. And we've got a serious problem with antibiotic resistance, and it's been in the news almost daily. The most recent scare has been with multi-drug resistant tuberculosis. Basically, this doesn't respond to any antibiotics. And this really is a sustainability issue, right? Antibiotics have revolutionized our quality of life and healthcare, and we want them to be sustainable and continue to use them. Unfortunately, resistance rates are increasing, but our discovery of new drugs is decreasing. So we have a sustainability problem there. And what we're bringing to the table is a different perspective of, okay, what can we do from an environmental standpoint? And this is in line with what the World Health Organization has called for, a global strategy to contain resistance. So if we can slow the spread of resistance, then our drugs should last for many more years to come. And in our research, we've been looking at the DNA as the environmental contaminant of concern. And so that's been a little bit different perspective from a traditional pathogen, but really makes sense in the case of resistance because DNA is shared among bacteria. That's a unique property. And it's especially common phenomenon with antibiotic resistance genes, that they're shared. So it's not good enough necessarily to just kill or contain bacteria, but we have to think about their DNA. And where does this fit in with water? Well, what happens when we consume antibiotics, right? They pass through our bodies, they go through sewage treatment, and they end up in waterways. And there have been some surveys by the U.S. Geological Survey showing drugs in our water. And the same thing we've done in our research group started saying, well, what about surveys of antibiotic resistance in our water? And we've really found some interesting and concerning trends. I want to give a little bit of background about this. We started off doing this research in Colorado in a watershed there, the South Platte River Basin. This has been a good model system because the source of the water is pristine in the Rocky Mountains, and then it goes through a gradient of urban influences. And I was working on this research when I first came to Virginia Tech, and it's kind of reached a crescendo this fall and had good synergy with this TSTS project. Basically, we mapped out wastewater treatment plants, the red dots, and animal feeding operations, the yellow triangles. I don't know how many of you are aware that actually most of our antibiotics in this country are administered to livestock, not people. So that's a big source of antibiotics to the environment. And what we found, which was astonishing even to us, we wanted to see if there's a correlation between upstream capacities of wastewater treatment plants and animal feeding operations and antibiotic resistance genes in the river. And yes, there is. In fact, a very strong one we saw an R squared of 0.92. So for any point in the river, you could predict the levels of resistance genes based on how many wastewater treatment plants, animal feeding operations are upstream, and their relative distances. That strengthened the model, and that's important because that tells us something about the importance of transport and getting the resistance genes there. And this is really important to see this quantitative relationship because there are a lot of naysayers. This is kind of an issue that, we've got a lot of problems and not everybody wants another problem to deal with, but we're trying to look at this positively, like, well, we're understanding this problem and what can we do about it. One thing is looking at wastewater treatment plants and improving treatment, and we did a parallel study actually supported by the NSFREU program here, paid for an undergrad who did a survey of purple pipes. So those are pipes that transport treated wastewater, so it's been treated to the highest standards, and then it's transported to used for irrigation. So this is one water sustainability approach. But we did the first study of a survey of antibiotic resistance genes in these pipes, and we saw that, yes, there is reason to potentially be concerned. There is growth of antibiotic-resistant bacteria, and so it's something that, where engineering fits in, like, okay, we're aware of this issue. What are some potential solutions? So this got quite a bit of press attention. So Flagstaff, Arizona is an area where they have water limitation issues, and they use recycled water. So they released our study, and then it was in the New York Times. And then it really kind of snowballed, literally, because they used the water that we tested to make snow. They just started this at the Snow Bowl near Flagstaff, Arizona. And this is kind of an unprecedented use of recycled water. And it was already controversial, so this definitely kind of fanned the flames. And there was also an article about this in the Nature News publication. And I'm actually going out to Flagstaff next week. So the positive side of this is I think the utility there has really tried to address this. And so they've called together an expert panel to say, okay, this is a problem. Thank you, scientists. Now give us some advice on what to do about it. Okay, so where does this fit in with the StREAM lab? We really need to start understanding mechanisms by which the antibiotic resistance genes are spreading. And so thanks to Cully and colleagues, we have this great model system here at Virginia Tech where we can really study closely what is it that's contributing to the spread of resistance genes. Is it sediment transport? Is it runoff? What role does manure play? We've also got an interdisciplinary research team, and again, this is a unique thing about Virginia Tech. We've got soil science experts, biological systems engineering. Kang Sia was just hired in crop, soil, and environmental sciences, and she's an expert at measuring the antibiotics in these complex matrices. That's challenging. Dairy sciences, Catherine Knowlton has been a great asset. So she's looking at, what happens when we feed cows with this antibiotic or that antibiotic, and she's been pretty passionate about it. So we're looking in the watershed, looking at baseline contributions of antibiotic resistance genes and bacteria. How are they distributed in the watershed, and how are they being transported? And then we want to create a basic mass balance model. And here's the lovely StREAM lab that Cully talked about. And we went out there and we mapped out the watershed and where overland flow occurs. There's not a lot of that. It has to be a certain kind of storm event. And we're also looking at where they apply manure. Now, this is a pretty impaired watershed, I guess is the term I've heard used. They've been applying animal manure forever. I think 100 years. Yeah. So, but it goes through cycles, and so they're actually applying this week. So we got a baseline before that application, and now we're going to see what happens after they apply the manure. And then we also have a forested area that's a good control. And then Catherine Knowlton's group is focusing on the cow and different kinds of antibiotics, and we've been working with Kang Sia and seeing the excretion rates of antibiotics, excretion rates of resistant bacteria. So in my lab, it's all about the DNA. We get these soil and manure samples, and the first thing we do is get the DNA out and see what's going on and quantify resistance genes. You can actually, again, think of them as contaminants. And what method do we use to quantify these contaminants? We use quantitative PCR. And this setup is all at ICTEST too. And then Kang Sia's group uses LC-MS to look at the antibiotics. And then this is some of our results so far. And so, we're interested in, is it season that's a driver of the level of resistance genes? Or is it location in the watershed? And it looks like it's a little bit of both so far, which corresponds well with what we've seen in Colorado. And so right now we're focusing on getting out there after this current manure application. We're also working on developing some new next generation methods. So we want to use metagenomics, and actually we have a spin-off of that called plasmidomics, where you can just directly interrogate the genes that are present, including antibiotic resistance genes. Whereas with quantitative PCR, you kind of have to guess which genes you want to look at. This will just let us go straight and see what's there. And focusing on plasmids will help us capture resistance genes because they tend to aggregate on plasmids. And then we've got the excretion study. And then we're also interested specifically in mechanisms of DNA persistence and decay. So if DNA is the contaminant, we have some learning to do in terms of how it behaves. It's a biopolymer, and we just got funding from the Water Environment Research Foundation to start looking at that. And then technologies for removing DNA from wastewater effluents. So we've looked at UV and just published a study on that, and you think, wow, UV should be great because UV damages DNA. That's what it does. And it seems like it's kind of temporary damage that the microbes can recover and grow downstream. And UV doesn't leave a disinfectant residual. So if you're in a purple pipe for recycled water, after the UV's done, there's plenty of opportunity to regrow in the pipes downstream. We did a study on membrane removal, and that was actually pretty promising. So again, DNA is a unique contaminant. It's a long polymer. There haven't been much studies on how they behave in a membrane removal situation. And we found that actually you could get away with looser membranes because the DNA tended to aggregate with particles and be removed more easily than you would have predicted. So that was encouraging. And I mentioned I'll be in Flagstaff next week, and a lot of opportunities for proposals here because we've got the public health and water sustainability nexus coming together. We've had several publications in the last year. This one has just been accepted, and this focuses on what happens with recycled water in purple pipes. And here's the grant that was just funded by the Water Environment Research Foundation. I listed 3 that are in review, and then I remembered, oh yeah, there's actually a 4th that I forgot to list there, and that's one that Catherine Knowlton is leading to the Virginia Ag Council, again focusing on the excretion of antibiotics. And key collaborators, in addition to the Virginia Tech folks, we've had people at Colorado State. Mazdak Arabi's been instrumental in establishing this watershed framework that we're now trying out here at Virginia Tech. Diana Aga at University of Buffalo, and we had Fulbright Scholar for the summer as well.